The plant kingdom. In the previous chapter, we looked at the broad classification of living organisms under the system provided or proposed by Whittaker in 1969, wherein he suggested the five kingdom classification viz. Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we will deal in detail with further classification within kingdom Plantae, popularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of Monera protista having cell walls have now been excluded from Plantae, though earlier classification placed them in the same kingdom. So, the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperms, and angiosperms under Plantae. Let us also look at classification within angiosperms to understand some of the concerns that influence the classification systems. The earlier systems of classification used only gross superficial morphological characters such as habit, color, number, and shape of leaves, etc. They were based mainly on vegetative characters or on the androecium structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characters. Also, the artificial system gave equal weightage to vegetative and sexual characters. This is not acceptable since we know that often the vegetative characters are more easily affected by the environment. As against this, natural classification systems developed which were based on natural affinities among the organisms and considering not only the external features but also the internal features like ultrastructure, anatomy, embryology, and phytochemistry. Such a classification for flowering plant was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification systems based on evolutionary relationship between the various organisms are acceptable. This assumes that organisms belonging to same taxa have a common ancestor. We now use information from many other sources too to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Numbers and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome numbers, structure, behavior, and chemotaxonomy that uses the chemical constituents of the plants to resolve confusions, are also used by taxonomists these days. Algae are chlorophyll-bearing, simple thyroid, autotrophic, and largely aquatic, both freshwater and marine organisms. They occur in a variety of other habitats, moist stones, soils, and woods. Some of them also occur in association with fungi, lichen, and animals. Example on sloth bear. The form and size of algae is highly variable, ranging from colonial forms like volvox and filamentous forms like Ulithrix and Asparagera. Figure 3.1. A few of the marine forms such as scarabs form massive plant bodies. The algae reproduce by vegetative, asexual, and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the most common being the zoospores. They are flagellated, motile, and on germination give rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Ulithrix, and non-flagellated, non-motile but similar in size as in Sparagara. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in the species of Eudornia is termed as isogamous. Fusion between one large non-motile static female gamete and a smaller motile male gamete is termed as zoogamous. Example in Fucus and Volvox. Algae are useful to man in variety of ways. At least half of the total carbon dioxide fixation on Earth is carried out by the algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are the paramount importance as primary producers of energy-rich compounds which form the basis of the food cycles of all aquatic animals. Many species of Porphyra, Laminaria, Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produces large amount of hydrocolloids, water-holding substances, example algae, brown algae, and carrageenan red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Gracilaria, are used to grow microbes and in preparation of ice creams and jellies. Chlorella, a unicellular alga rich in proteins, is used as food supplement even by space travelers. Excuse me. The algae are divided into three main classes: chlorophyceae, phaeophyceae, and rhodophyceae. Chlorophyceae, the members of chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial, or filamental. They are usually grass green due to the dominance of pigments chlorophyll A and B. The pigments are localized in definite chloroplasts. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage bodies called paranoids located in the chloroplast. Paranoids contain proteins besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by egg fragmentation or by formation of different types of spores. Asexual spores is by flagellated zoospores produced in zoosporangia. The sexual reproduction shows considerable variation in the type and formation of the cells in it. Maybe isogamous, anisogamous, or oogamous. Some commonly found green algae are Chlamydomonas, Vox, Ulothrix, Spirogara, and Chara. Phaeophyceae. The members of Phaeophyceae or brown algae are found primarily in the marine habitats. They show great variation in size and form. They range from simple branch filamentous form ectocarpus to profusely branched forms as represented by gills, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids, xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of the xanthophyll pigment fucoxanthin present in them. Food is stored as complex carbohydrates, which may be in the form of laminarin or mannitol. The vegetative cells have a cellulosic wall usually covered on the outside by a gelatinous coating of algin. The protoplast containing, in addition to plastids, are centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has a stalk, the stipe, and a leaf-like photosynthetic organ, the frond. The vegetative reproduction takes place by fragmentation. Asexual reproduction in most brown algae is by flagellate zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the Ogonium oogamous species. The gametes are pyriform, pear-shaped, and bear two laterally attached flagella. The common forms are ectocarpus, dictyota, laminaria, sargassum, and fucus. The members of the Rhodophyceae are commonly called red algae because of the predominance of the red pigment or phycoerythrin in their body. Majority of the red algae are marine with a greater concentration found in the warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depth in oceans where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization development. The common members are Polysiphonia, Porphyra, Gracilaria, and Gelidium. Let's get back to the table. Chlorophyceae class is having Chlorophyceae, common name is green algae. Major pigments of Chlorophyceae is chlorophyll A and B. Food is stored in the Chlorophyceae in the form of starch. Cell wall of Chlorophyceae is cellulose. The flagellar number and position of insertions is 2 to 8, equal and apical. And Chlorophyceae is having habitat of fresh water, brackish water, and salt water. Phaeophyce is brown in color, and that's why it's said to be brown algae. Now, Phaeophyce is having chlorophyll A, C, and fucoxanthin as its major pigments. Phaeophyce is having mannitol, laminarin, and the cell wall of Phaeophyce is made up of cellulose and algin. The Phaeophyce is having two flagell number, and the position is unequal, and insertions are lateral. Phaeophyce is having habitat of freshwater, rare brackish water, salt water, and then we move on to rhodophyce. Rhodophyce is red in color, and it's having red algae. Major pigments of rhodophyce is chlorophyll A, D, phycoerythrin. Rhodophyce is having stored food as Floridian starch. Rhodophyce is having cell wall as cellulose, pectin, and polysulfate esters. Rhodophyce is having flagellar number and position of insertions as the position is absent. Rhodophyce is having habitat of freshwater. Some are in freshwater, brackish water, salt water, and mostly are found in salt water. Moving on to bryophytes. Bryophytes 3.2. Bryophytes include the various mosses and liverworts that are found commonly growing in moist shaded areas in the hills. Archegoniophore, gamma cup. Okay. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in plant succession on bare rocks or soil. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect. And and attached to the substratum by unicellular or multicellular rhizoids. They lack true roots, stem, or leaves, but they may possess root-like, leaf-like, or stem-like structures. The main plant body of the bryophyte is haploid. It produces gametes, hence is called a gametophyte. The sex organs in bryophytes are multicellular. The male sex organ is called entheridium. They produce biflagellate entherozoids. The female sex organ called archegonium is flask-shaped and produce a single egg. The entherozoids are released into water where they could come in contact with archegonium, and an entherozoid fuses with the egg to produce the zygote. Zygotes do not undergo reduction division. Immediately, they produce a multicellular body called a sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division meiosis to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds, and other animals. A species of sphagnum, a moss, provide peat that have long been used as fuel and as packing material for transhipment of living materials because of their capacity to hold water. Mosses, along with lichens, are the first organisms to colonize rocks and hence are of great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mounds on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. 3.2.1 Liverworts. The liverworts grow usually in moist, shady habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of liverwort is thalloid. Example: Markentia. The thallus is dorsiventral and closely appressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverworts takes place by fragmentation of thalli or by the formation of specialized structures called gamma, singular gamma. Gamma are green multicellular asexual buds which develop in small receptacles called gamma cups located in the thalli. The gamma become detached from the parent body and germinate to form new individuals during sexual reproduction. Male and female sex organs are produced either on the same or on the different thalli. These sporophyte is differentiated into a foot, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophytes. 3.2.2 moss is the predominant stage of the life cycle of moss is the gametophyte, which consists of two stages. The first stage is the protonema stage, which develops directly from a spore. It is a creeping, green, branched, and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright, slender axes bearing the spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoid. This stage bears its organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, these sex organs, Ncert and archegonia, are produced at the apex of the leafy shoots. After fertilization, the zygote develops into a sporophyte consisting of a food, seta, and capsule. The sporophyte in mosses is more elaborate than in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum, and Sphagnum. Pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medical purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to process vascular tissues, xylem and phloem. You shall study more about these tissues in chapter 6. The pteridophytes are found in cool, damp, shady places. Through some may flourish well in sandy soil conditions. You may recall that in bryophytes, the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophytes, the main plant body is the sporophyte, which is differentiated into true root, stem, and leaves. Figure 3.3. These organs possess a well differentiated vascular tissues. The leaves in pteridophyta are small microphylls as in Slachinella, or large microphylls as in ferns. The sporophytes bear the sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, the sporophylls may form distinct compact structures called strobili or cones. Laginella equisetum. The sporangias produce spores by meiosis in the spore mother cells. The spores germinate to give rise to inconspicuous, small but multicellular free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of the living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organs called Ncert and archegonia respectively. Water is required for transfer of anthrazoids. The male gametes released from the Ncert to the mouth of the archegonium. Fusion of male gametes with the egg present in the archegonium results in the formation of zygote. Zygote thereafter produces a multicellular, well-differentiated sporophyte, which is the dormant phase of the pteridophyte. In majority of the pteridophytes, all the spores are of similar kinds. Such plants are called homosporous. Genera like Laginella salvinia, which produces two kinds of spores, macro large and micro small spores, are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophytes. Respectively, the female gametophytes in these plants are retained on the parent sporophytes for variable periods. The development of the zygotes into young embryos take place within the female gametophytes. This event is a precursor to seed habit, considered an important step in evolution. The pteridophytes are further classified into four classes: Psilopsida, Psilotum, Lacopsida, Slachinella, Lycopodium, Sphenopsida, Equisetum, and Pteropsida, Triopteris, Pteris, Adiantum. 3.4 Gymnosperms. The gymnosperms, naked sperma seeds, are plants in which the ovules are not enclosed by any ovary wall and remain exposed. Both before and after fertilization, the seeds that develop post-fertilization are not covered, that is, are naked. Gymnosperms include medium-sized trees or tall trees and shrubs. 3.4 One of the gymnosperms, the giant redwood tree Sequoia, is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association in the form of mycorrhiza, pinus, while in some others, cycas is small specialized roots called coralloid roots are associated with nitrogen-fixing cyanobacteria. The stems are unbranched cycas or branched pinus citrus. The leaves may be simpler or compound in cycas. The pinnate leaves persist for a few years. The leaves in gymnosperms are well adapted to withstand extreme of temperatures, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. Their thicker and sunken stomata also help to reduce water loss. The gymnosperms are heterospores. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophylls, which are arranged spirally along an axis to form lax or compact strobili or cones. The strobili bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspores develop into male gametophytic generation, which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called pollen grain. The development of pollen grains take place within the microsporangia. The cones bearing microsporophylls with ovules or megasporangia are called microsporangiate or female strobili. The male and female cones of strobili may be born on the same tree pinus. However, in cycas, male cones and megasporophylls are born on different trees. The megaspore mother cells is differentiated from one of the cells of the nucellus. The nucellus is protected by envelopes, and the composite structure is called an ovule. The ovules are born on megasporophylls, which may be clustered to form the female cones. The megaspore mother cells divides mitotically to form 4 megaspores. One of the megaspores enclosed within the megasporangium develops into a multinuclear female gametophyte that bears 2 or more archegonia or female sex organs. The multinuclear female gametophyte is also retained within megasporangium. Unlike bryophytes and pteridophytes in gymnosperms, the male and female gametophytes do not have an independent free-living existence. They remain within the sporangia, retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules born on megasporophylls. The pollen tube carrying the male gametes grows towards archegonia and the ovules and discharge their contents near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and the ovules into seed. These seeds are not covered. Angiosperms. Unlike the gymnosperms where the ovules are naked, angiosperms are flowering plants. The pollen grains, the ovules are developed into specialized structures called flowers. In angiosperms, the seeds are enclosed in fruits. The angiosperms are exceptionally large group of plants occurring in wide range of habitats. They range in size from the smallest wolfia to tall trees of eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicines, and several other commercially important products. They are divided into two classes, the dicotyledonous and the monocotyledonous. The dicotyledons are characterized by seeds having two cotyledons, reticulate venation in leaves, and trimerous or pentamerous flowers, having 4 or 5 members in each floral whorls. The monocotyledons, on the other hand, are characterized by single cotyledonous seeds, parallel venation in leaves, and trimerous flowers having 3 members in each floral whorls. The male sex organs in the flower is the stamen. Each stamen consists of a slender filament with an anther additive. Within the anther, the pollen mother cells divide by meiosis to produce microspores, which matures into pollen grains. The female sex organs in a flower is the pistil. Pistil consists of a swollen ovary at its base, a long slender style and stigma. Inside the ovary, ovules are present. Generally, each ovule has a megaspore mother cell that undergoes meiosis to form 4 haploid megaspores. 3 of them degenerate and 1 divide to form the embryo sac. Each embryo sac has 3-celled egg apparatus, 1 egg cell and 2 synergids, 3 antibody cells and 2 polar nuclei. The polar The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grain, after dispersal from the anthers, are carried by wind or various other agencies to the stigma of the pistil. This is termed as pollination. The pollen grains germinate on the stigma, and the resulting pollen tubes grow through the tissue of stigma and style and reach the ovule. The pollen tubes enter the embryo sac where two male gametes are discharged. One of the male gametes fuses with the egg cell syngamy to form a zygote. The other male gamete fuses with the diploid secondary nucleus to produce triploid primary endosperm nucleus, or pen. Because of the occurrence of two fusions, that is syngamy and triple fusion, this event is termed as double fertilization, an event unique to angiosperms. The zygote develops into an embryo with one or two cotyledons, and the pen develops into the endosperm, which provides nourishment to the developing embryo. The ovules, the synergids and antibodial regenerate after fertilization. During these events, the ovules develop into seeds and ovides develop into fruits. The life cycle of an angiosperm is shown in figure 3.6. Plant life cycles and alternation of generation. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies. The haploid plant body produces gametes by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. These in turn divide by mitosis to form haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generation between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals representing them differ in the following patterns. The sporophytic generation is represented only by the one-celled zygote. There are no free-living sporophytes. Meiosis in the zygote results in the formation of haploid spores. The haploid spores divide mitotically and form the gametophyte. The dominant photosynthetic phase in such plant is the free living gametophytic. This kind of life cycle is termed as haplontic. Many algae such as volvox, Spirogyra, and some species of chlamydomonas represent this pattern. On the other extreme is the type wherein the diploid sporophyte is the dominant photosynthetic independent phase of the plant. The gametophytic phase is represented by the single to few celled haploid gametophyte. This kind of life cycle is termed as diplontic. Alga fucus species represent this pattern. In addition, all seed-bearing plants, that is gymnosperms and angiosperms, follow this pattern with some variation wherein the gametophytic phase is few to multicelled. Bryophytes and pteridophytes interestingly exhibit an intermediate condition, haplodiplontic. Both phases are multicellular, however they differ in their dominant phases. A dominant independent photosynthetic thyroid or erect phase is represented by a haploid gametophyte, and it alternates with the short-lived multicellular sporophyte, totally or partially dependent on the gametophyte for anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant independent photosynthetic vascular plant body. It alternates with multicellular sporophytic autotrophic independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycle. All pteridophytes exhibit this pattern. Interestingly, while most algal genera are haplontic, some of them, such as Ectocarpus polysyphonia, kelps, are haplodiplontic. Fucus as an alga and is diplontic. Okay, summary. Plant kingdoms includes algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Algae are chlorophyll-bearing, simple thyroid and autotrophic and largely aquatic organisms. Depending on the type of pigment possessed and the type of stored food, algae are classified into three classes, namely chlorophyceae, phaeophyceae, and rhodophyceae. Algae usually reproduce vegetatively by fragmentation, asexually by formation of different types of spores, and sexually by formation of gametes, which may show isogamy, anisogamy, or oogamy. Bryophytes are plants which can live in soil but are dependent on water for sexual reproduction. Their plant body is more differentiated than that of an algae. It is thallus-like and prostrate or erect and attached to the substratum by rhizoids. They possess root-like, leaf-like, and stem-like structures. The bryophytes are divided into liverworts and mosses. The plant body of liverworts is thalloid and dorsiventral, whereas mosses have upright, slender axis bearing spirally arranged leaves. The main plant body of a bryophyte is gamete producing and is called a gamete. It bears a male sex organs and called enteridia. The female sex organs called archegonia. The male and female gametes fuse to form zygote, which produces a multicellular body called a sporophyte. It produces haploid spores. The spores germinate to form gametophytes. In pteridophytes, the main plant is a sporophyte, which is differentiated into true root, stem, and leaves. These organs possess well-differentiated vascular tissues. The sporophytes bear sporangium, which produces spores. The spores germinate to form the gametophytes, which require cool, damp places to grow. The gametophytes bear male and female sex organs called enteridia and archegonia, respectively. Water is required for transfer of male gametes to archegonium, where zygote is formed after fertilization. The zygote produces a sporophyte. The gymnosperms are the plant in which ovules are not enclosed by any ovary or ovary wall. After fertilization, the seeds remain exposed, and therefore these plants are called naked seeded plants. The gymnosperms produce microspores and megaspores, which are produced in microsporangia and megasporangia borne on the sporophylls. The sporophylls, microsporophylls, and megasporophylls are arranged spirally on axis to form male and female cones, respectively. The pollen grain germinates in pollen tubes, releases the male gamete into the ovule, where it fuses with the egg cell in archegonia. Following fertilization, the zygote develops into embryo and the ovules into seeds. In angiosperms, the male sex organ stamen and the female sex organ pistil are born in a flower. Each stamen consists of filament and an anther. The anther produces pollen grains, male gametophyte after meiosis. The pistil consists of an ovary enclosing one to many ovules. Within the ovule is the female gametophyte or embryo sac, which contains the egg cell. The pollen tube enters the embryo sac, where two male gametes are discharged. One male gamete fuses with egg cell, syngamia, and the other two fuses with diploid secondary nucleus, triple fusion. This phenomenon of two fusion is called double fertilization and is unique to angiosperms. The angiosperms are divided into two classes, the dicotyledons and the monocotyledons. During the life cycle of any sexually reproducing plant, there is an alternation of generation between the gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals may show different patterns of life cycles: haplontic, diplontic, or intermediate. So, guys, that was our reading of the chapter. Plant Kingdom. I hope you liked it. I am very much open to suggestions. Please do not write it down in the comment section, and I'll seriously be noting it down and will be abiding according to that. Goodbye. Please subscribe to the channel.